Hello everybody, welcome again to the webinar for batch processing with Nexcelom datasets, moving from data to results with a single click with FCS Express. Many Nexcelom customers may be surprised to know that FCS Express will allow you to quickly export results from the K2, CBA Vision, Celigo, and other instruments directly to PowerPoint, PDF, and Excel at the same time across many samples with a single click. In this webinar, we will be covering how to effectively use the data list, batch processing for your Nexcelom derived datasets in FCS Express to accelerate the process to getting you results. Using batch processing in FCS Express is easy to do, and it will help eliminate bottlenecks in your analysis and pipeline. As a little overview, the current status of when you acquire data from a Nexcelom instrument is that it will launch FCS Express, which is very easy. So at the end of your acquisition run, you press the button to launch FCS Express. But when you're acquiring many data sets, many instances of FCS Express will open. There'll be one instance or one layout for every single file, which makes it a little bit bulky to process, organize, and to compare results between different files. So the solution with FCS Express is batch processing, which allows quick analysis of many files with a single click. You can combine data from many experiments into one consolidated report, and you can export all of that data to one Excel document, to a PowerPoint, to a PDF, really in whatever format you like. So today we're going to cover how to learn to save existing launched layouts from your Nexcelom instrument in FCS Express for reuse with batch processing. We're going to learn how to add multiple .nxstat files, which are the file type for Nexcelom data files, to the data list. And we're going to set up and run a batch process to export to Excel and PowerPoint. So to get started, let's switch over to FCS Express and take a look at what the export formats currently look like for Nexcelom datasets. So what you see up on the screen here is a example of a data file in AOPI .nxstat file. You can see here in the title of the Nexcelom layout that was created for FCS Express. That's what this experiment is. And in our data list, we have one data file. So when you click on the button in the Nexcelom software to launch FCS Express, this is what you get. Now, if you have multiple data files, what will happen is that FCS Express is going to launch up multiple times. So you can see here that I've actually acquired two data files today, and I have separate instances of FCS Express showing the data files and the reports for this. So as you can imagine, this can get a little bit bulky, especially if you're working with more than two data files. I know most people acquire many data files when analyzing this data. So the solution is going to be essentially adding data files to what we call our data list here in FCS Express, resaving this layout, and enabling batch process. So to get started today, the first thing I'm going to show you is how you can add some additional data files to the data list and how this works. So what I'm going to do is pop the data list out here. If it was docked in the layouts that you're using, you might see it kind of pinned over here to the side, but you can pull that out at any time. And what we can do in FCS Express is if we want to work with different data files within the same analysis template, so working with these same plots and statistics, we can click on the plus button in the data list and click on Add Data File. Now when you do this, you're going to be brought to your Windows navigation screen, and you're going to be prompted to go get a data file. So one important thing here is that when you do this for the first time, it may not be set to Nexcelom files in this file of type list. It may actually be set to FCS files. So the first thing of note is make sure that you choose Nexcelom.nxstat files. And when you do so, you can click on one or many or multiple select data files and add them to the data list, and you can see that they become available in the data list for use. So when we do this, if we click on the iteration number here, you can see that in our layout to the left, the plots and the statistics are all updating with the new data files that you've clicked on from the data list. Additionally, the data list or the data files that are showing in this layout can be managed by clicking on the Data tab. And you'll notice that there's buttons here for previous and next change data on all objects. So what that will do is as I click on those buttons, you can see that we're going to iterate through the data files in the data list. And when I say iterate, I simply mean change between the data files. So you can work with either of these tools. If you need to quickly jump between data files that are far apart, you can click on the data list. If you need to just use next and previous, you can simply use the buttons that are located in the data tab. So that's a quick primer about how to add data files to the data list and essentially work with the layouts that you currently have. But when you create these layouts from the Nexcelom instrument, they get created and saved with a file name like this. It will say APO intensity 2. It will probably have the name of the data file within the layout name. So we don't necessarily want to overwrite this. What we want to do is create a new FCS Express layout, a new .fa file that we can use over and over again for batch processing. So this is quite simple to do. Essentially, you can load up any template that you've worked with your Nexcelom data in the past. If you've launched up a brand new template, you can use that. But what we're going to do is click on the File tab, and we'll do a Save As. And when we do a Save As, what we're prompted to do here is go in and save this layout file with a new name. So I'm going to call it AOPI. I'm going to put underscore, I'm going to put batch processing layout, and we'll simply just give it a new name. So what that will do is it will not overwrite what you already have done in the past. It will create a new instance of that. So that way, when we need to come back and analyze or batch process data files in the future, we don't have to just load up all of the individual layout files that were created for individual files in the past. We can simply come back in. You can see here I'm going to use the handy recent layouts button here within the FCS Express startup screen. Alternatively, you can click on the File tab and click Open, and you can launch it up. And you can see that it's going to launch up with whatever you had done in the past, the same layout. Now, the other way that this will work is in these layouts that are created. Essentially, these have been defined as one tube panels. And really, what that means is it allows you to clear out everything that's in the data list. All of your plots will be blank. But if we add back another data file from another experiment or from whatever it is we want to do for that day, it's going to automatically populate into the plots of interest. So keep this in mind when you have these templates saved for use. If you had some data files that were in the data list previously, you can simply clear them out, and then you can load in any additional data files you want. So again, you can start seeing that it's becoming a little bit more convenient to work with sets of data, to work with sets of folders of experiments of data, and to navigate between them quickly and easily. But in the end, what we want to do with these data sets is quickly and easily generate some sort of report. So generally, folks will export to PowerPoint, they'll export to PDF, they'll export results to Excel, and that's where your final reports are going from this analysis once you've loaded up your data and you've set your marker and gate positions. Now, in the past, you may have been clicking on the File tab, Export, and exporting each individual data file one by one as a PDF or a PowerPoint. You may have been copying and pasting results from any of these tables out to Excel, but we're going to make that much easier, and we're going to make it so that you can do that across any number of data files very quickly and efficiently. And we do that in FCS Express through batch processing, which is located in the Batch tab. And what we're going to do is click on Batch Actions. Now, what batch actions are going to do are they're going to allow us to define a series of different actions, such as save to PDF, save to PowerPoint, save to Excel, and then repeat that action over and over again for as many data files as you may have in the data list that day. So to get this set up and to get started, I'm going to run a few exports today, primarily to show you how they all work. I'm going to click on Save to PDF. And what's going to happen is FCS Express is going to launch up a Create Export to PDF action. And a few important points here. One is when you're saving PDFs, the default is to save a separate PDF for every data file. That's absolutely fine if you want to do that. the other option that you have here that I generally prefer to choose is combine the output from all iterations or all data files into one PDF. So in this instance, I have 7 data files in my data list, so that will create one PDF with 7 pages instead of 7 PDFs, 1 page each. And to implement this, I'm going to click on Save to a new file. We're going to export this out to my folder, and we're going to call this today's analysis. We'll click Save and click OK. So what will happen now is when I run the batch process, all 7 of my data files are going to be exported to one PDF file. Now additionally, if you wanted to do the same thing for something like PowerPoint, we can choose Save to PowerPoint. You can see that in these batch processing options, the default here is to load everything into the same PowerPoint presentation. So we'll have one PowerPoint presentation with 7 slides, 1 slide for every data file. And again, I'm going to indicate a place to save this. We're going to call this again today's analysis, and we'll click Save. So just like that, we've set up 2 export options, one export to PDF and one export to PowerPoint. But again, those are more like static images, right? Those are images of what's on the screen, a report, which is here. And sometimes you do need to have numeric data associated with many data files and exporting that to things like Excel or Prism or CSV files. So we make this very simple in FCS Express by using the Export to Excel column mode feature. Now, when we click on this feature, it's going to prompt us to save to a new file. And essentially what that's asking us to do is save to some sort of Excel document. And it's okay if we don't have an Excel document created yet. We'll create one. We'll call it, again, today's analysis. We'll click Save, and we'll click OK. Now what's going to happen here is FCS Express creates this Excel document, but we do not have any information associated with that yet. We don't know what to export to Excel. Do we want to export things like the file name, the assay description, the date, statistics? In fact, we probably want to export all of those things, a lot of the information that's here. And the way that we populate an Excel export in FCS Express through batch processing is generally through a drag and drop. So a lot of the information you want to export is already in your layout, so things such as the file name. So if I select the file name and I drag this into my Excel column export, you can see that the first column that's going to be located in Excel is going to be the file name. So there'll be one column for file name, and in this batch run there'll be 7 rows with each of the file names for all of the data files. So additionally, I may want to put in things like today's date or the analysis date. I can drag and drop that in there. And importantly, if I have statistics on my layout that I want to populate, I can drag and drop them in there as well. So for instance, the percentage of FL1 intensity, I can drag that text box up here, and it's going to end up as a column. Additionally, if I wanted to grab from here, I can do that as well. Now, FCS Express does have some advanced functionality for selecting statistics associated with different things on your plots. So if I know I just want to grab a statistic that's associated with this marker, perhaps I don't want the percent, but if I drag and drop that in there, FCS Express is going to launch up a dialog that asks you which statistic from M1 marker do you want. So if I wanted to put in something like the number of events and perhaps the median of that marker, I can select one or multiple or everything that's in here. I can click OK. And those 2 statistics, the number of events and the median for the FL2 intensity marker, are going to appear. Now additionally, if you have a statistics box or you have a text box shown in the layout that has a number of different statistics and you know that you may want to choose all of them, you can simply grab the entire text box. And anything that's in there that's a statistic, anything in here you can see we have percentage of gated cells in the concentration, is going to be populated into your Excel mode export. So again, a very quick and easy way to get information there is if you find a text box that has some statistics in it. Here we have the total cell count and the dilution factor. You can drag and drop that in there. So one thing, again, to take away from this is any of the statistics that you'd like, you can simply drag and drop them into your Excel mode export to populate this and get it running. So we've spent a little bit of time now putting together these exports, and the good news is we don't have to do this again. So if I click Save in this layout, the next time that I come back to this layout, whether it be with this data set or a new data set, I don't have to populate what's been happening in these batch processing actions again. I can simply come in here, I can change the name of the file, right? I may not want to call this today's analysis again because I may want to use a better descriptor of when this data was created. I can change the data file name and I can run this. But let's take a look at what running this will do. So I can close the batch actions now because I'm ready to run. Generally what you want to do is before you run the batch actions, you want to go through and make sure that all of your marker and gating positions are correct. And I'm going to just adjust this one marker a little bit. And we can click run. And now when I click run, FCS Express is going to perform three actions. It's going to export to PowerPoint, it's going to export to PDF, and it's going to export to Excel. You can see in the background here, it's changing data files, and it's doing that across all the data files that are been loaded into the data list. And if this is your full experiment, it's everything that's associated with that experiment. So what we're going to see now is Microsoft Excel is going to launch up, PowerPoint is going to launch up, and in each of these, you notice that we have a column associated with all of the statistics that we chose to export, things like the file name and the intensities and the keywords. We have a PowerPoint presentation that was created over here, and on every PowerPoint slide, we have a new data file. Now, the nice thing about this too is when you're working with PowerPoint exports from FCS Express, all of the objects on the page are high resolution vector graphics. That means I can grab an individual plot, I can resize it, I can move it. If I need to grab this for a publication, I can make it very large and very high resolution. Even things like text boxes can be moved around within these. So it's very convenient to have these data file exports within that format. Now additionally, we have a PDF file that was created, and you can see here it's a 7-page PDF file, and there's one page for all of the analysis that was exported as part of this batch process run. So what I'm going to do now is kind of walk back a step here and again mock up what this will look like a little bit if you are going to be using this layout over and over again. Because again, what we did here in the beginning, we did a save as, we set up all of our batch process, we added some data files to the data list. But if you come back tomorrow or you have another data set of your Acridine Orange data files that you need to analyze, you can simply close this layout. And the next time I come in and I use FCS Express, I can open up this batch processing template that we've created. FCS Express is going to load it up, and it's going to have, again, the last data files that you had analyzed in here, and that's fine. And if you wanted to, you could even clear these data files out before you save it. But the process here would be I come in and I remove the data files from last week's experiment. I go in and I find some new data files from this week's experiment. Grab maybe these ones down here at the bottom of my list and click open. Again, I'm going to come in and review that my markers are set in all the correct positions. I'm going to check in my batch actions here that I'm saving with a new file name. So in this case, I'm just going to let it overwrite the existing file name. That's fine for this webinar. But if you wanted to, you could also block overwriting existing files, which is kind of a good option to use to make sure that you don't overwrite what you've already done. But you can see here in just, maybe about 20 or 30 seconds, we've loaded up data files, we've reviewed what's going on with the markers, everything's set in the correct positions, we've set our settings in the batch, and we're going to click run. And again, what we're doing now is we're exporting the data for everything that's in the data list. We're getting that exported to PowerPoint, PDF, and Excel. And you can see I actually made a mistake here on purpose. And this is one of the kind of common pitfalls that folks will run into, is that when you run the batch process, it's going to run from whatever data file you happen to be on at the time. So I happened to be on iteration number 7, which was the last in the list. So what happened here in the batch process is it tried to run data file 7, it tried to move to the next one, there is no next one, so the batch ended. So it's one thing to be careful of when you run batch. Make sure you, before you run the batch, make sure you click on the first iteration of the data list. Then when you click run, it's going to iterate across everything else that's in that data list. And again, this can be 7 files, it can be 70, 700, 7,000 even. It doesn't matter how many you have, as long as you can get those into the data list. The batch process is going to run these very quickly and efficiently and collate all of your results amongst all of your reports any way that you'd like. So one other functionality in batch processing I should discuss, or two, is one is if you don't happen to want to run an action that day, you can unselect it. So a very common thing is with printing, if I add a print action, I may want to print all these pages off into a hard copy report from my printer. If I want to save some paper that day, if I want to be kind to the environment, essentially what I can do is before I run this batch process, if I uncheck the print button, then this print action would not be run. Right? So if sometimes I want to print a report, sometimes I don't, I can choose to check or uncheck that. If I don't need a PDF report today, if I don't need an Excel export today, I can check or uncheck these actions to get that running. Now, additionally, if you want to review your gates on the fly, if you want to review your marker positions and make changes while the batch process is running, so it's another kind of convenient way to analyze data. So you can imagine you load up data files in the data list, you hit run on the batch process, it's kind of crunching through things. But if you wanted to move markers rather than reviewing all of the data files beforehand, there's a series of options associated with the batch. You can see here there's a little options button. And when we open up the options, we can choose an option called Unconditionally Pause between iterations. Now what this will do is when I run the batch process, it's going to use whatever I have set up on the layout for the first data file. It's going to move to the next data file and it's going to pause, and it's going to allow us to adjust any sort of markers or gates that we have, and it's going to allow us to do this on the fly. So you can see here we've moved to iteration number 2, and now the batch process has paused. And if I choose to come in here and change to move my marker position here, I can choose to continue and keep the changes, and it's going to move to the next data file. And what I'll be able to do at this point is move the marker again, choose to move on, continue, and keep that change. Now the other option I can do is if I want to have this marker stay in the same position for the rest of the data files, I can say restore the state of the layout before the next iteration. I can click continue, and you can see that it's actually going to move the marker position back to where it was beforehand. So here what I'm going to do is if I want to keep the change for everything else, I'll simply click run to end, and you can see that the batch process is going to run to the end of the batch processing list in the data list. It's going to complete our exports, and the batch process is going to complete. So here are my PowerPoint reports. I actually ran my print options today, so you may hear my printer in the background trying to run off a few print copies of that, so you get to hear that that works as well. And what we'll notice, again, in my options, we exported the PowerPoint, we ran a print, and the batch process has actually completed. So again, I hope you learned some information from this kind of quick webinar today. Again, the idea with this webinar is to show you some of the new tools in FCS Express, or some of the tools that they've actually been here for a little while that are going to provide some solutions to help you quickly analyze many data files with a single click. And essentially that single click is once you set up the batch processing in FCS Express and you resave that layout as a new template, you can load your data files into it, you can run the batch by clicking the run batch button. This also allows you to combine data files from many experiments into one consolidated report. So if you need one PDF with 7 pages from all of 7 of your data files, if you want to have 700 pages, or if you even want 700 individual PDFs, you can do that as well. Importantly, it can also export all of that data to one Excel file. So if you need those numeric results for downstream analysis or record keeping of this data, you can simply export all that information to Excel. And importantly, you can save that layout for quick access and convenience in the future. So anytime you have a similar experiment that you need to run, you go and draw up that template, you can click run on the batch process, and you'll be all set. So thanks again for joining us on the webinar today. We are going to take a few moments for questions after the webinar. We'll take a few moments to look through if anything has come through the questions and answer window. And again, thanks for your time today. If you have any questions in the future, please contact us at support@denovosoftware.com, and feel free to visit our website at denovosoftware.com at any time. Thanks again and have a good day.